हेलो स्टूडेंट्स, तो आज हम करेंगे चैप्टर बायोमोलिक्यूल्स. दिस इज वन ऑफ द वेरी इंपॉर्टेंट चैप्टर फॉर योर कंपटीशन point of view because a number of questions can be asked from this topic. This topic is easy for medical students but not for the non-medical students. For medical students, I feel that this chapter is on their fingertips, but non-medical students always find it difficult to learn something about the biomolecules. But whether you are a medical student or non-medical student, you must have some knowledge about molecules which are present in the living system, because a lot of chemistry is involved. As you are doing this, you are a science student and you are doing chemistry, so you must learn. Okay, so let's start with biomolecules. In the word biomolecules, bio means living. So molecules which are present in the living system, they are called the biomolecules. Molecule itself are not living, okay, but they are organized in such a way that in nature they ultimately eventually led to a living life or a living entity. So that's why they are called the biomolecules. So we have many different types of biomolecules like carbohydrates, amino acids, proteins, nucleotides, RNA, DNA, etc. So today we will go one by one for each of these. So first let's start with the carbohydrates. So as names indicate, carbohydrates means hydrates of carbon. Okay, so their general formula we can represent as CX and water Y, making it hydrates of carbon. So let's say particular example, for example glucose, it has C6H12O6 and this formula fits into this one. So let's say put X is equal to 6, so we have C6, and then put Y equal to 6, we have H12 and O6. So this satisfies this formula as a hydrates of carbon in case of glucose. But with time, as more and more research was going on, scientists were doing more and more work, it was found that certain molecules don't obey this rule. For example, acetic acid molecule, if you see, this is also follow this formula, like carbon 2 C2, so we can put X equal to 2, and then hydrogen 4 and oxygen 2. If we put Y equal to 2 here, then we have H4 oxygen 2. So acetic acid molecule obeyed this formula, hydrates of carbon. But we know that from our experiences, this molecule is not a carbohydrate. It behaves as an acid and it has no characteristic feature which corresponds to the carbohydrate. So in other words, this definition, hydrates of carbon, somewhat followed for some molecules but not for the all. And eventually further, if we see, there is no such a compound where we can see hydrates of carbon, like only carbon is present and some water molecules are present. We don't see any such a compound in nature. So this definition was not as good as it should be, so that it can satisfy all the carbohydrates. So with experiences, we came to know that carbohydrates are those molecules in which many hydroxy groups are present, and other functionality is either aldehyde group or the ketone group. So we redefined carbohydrates as a polyhydroxy, means many hydroxyl group, plus either aldehyde or ketone group, means CHO or C double bond O should be present, or compounds which can give polyhydroxy aldehyde or polyhydroxy ketone after the hydrolysis. So those compounds will be called as carbohydrates. So next we have the classification of carbohydrates. First is sugar, non-sugar. Sugar are those which are sweet in taste and they are crystalline in form. For example, glucose, fructose. Non-sugar, which are amorphous in nature and they don't have a sweet taste and they don't crystallize. So those are called non-sugar. So for example, cellulose and starch. Further, we started classifying carbohydrates into monosaccharides, oligosaccharides and polysaccharides. This classification is called more systematically classification. We know that mono means one. Saccharides corresponds to again sugar unit. So molecules in which there is only one sugar unit, like glucose, galactose, so those are monosaccharides. Oligo means few. So when we have two or more than two units, those are called oligosaccharides. And further it may be di, tri, tetra, etc. In case of poly means many. So when we have like hundreds or thousands of units of saccharides, then we call it as polysaccharides. It's more like a polymer. And examples are again starch and glucose. Further, if we study more about monosaccharides, then we have aldoses and ketoses based on their functional group. Aldoses means it has aldehyde and ketosis means it has keto group, which we have already defined in our earlier definition of polyhydroxy aldehyde and polyhydroxy ketone. So if aldehyde group is present, aldoses; if ketone group is present, ketosis. Now there may be 1 carbon, 2 carbon, 3 carbon like that. On the basis of that we can say aldotrioses, aldotetraoses, aldopentaoses. Similarly, keto again 2, 3, 4 carbon, we can say ketotriosis, ketotetrosis, ketopentaoses. So this was another way of classifying these. Now let's see the structure of carbohydrate, and this is the important topic and question can be asked from structure. The simplest carbohydrate which we have is glyceraldehyde. Glyceraldehyde. Okay, and then we have here glucose. Here we have galactose, and here we have fructose. Okay, so now let's see. In case of glyceraldehyde, carbon atom carrying the hydroxyl group next to the CH2OH is on right hand side. So in other words, we can say hydroxyl group which is attached to the carbon next to CH2OH is on right hand side. We call this as a D-glyceraldehyde. In case of glucose, let's draw the full structure. Right hand side OH, right hand side OH, right hand side H, right hand side OH. We have H, we have OH, we have H, we have H. Okay, so this is also D-glucose. Why? Because OH is on right hand side with respect to this CH2OH. In other words, OH which is attached to the carbon next to CH2OH is on right hand side. So we call it as a D-glucose. In case of galactose, let's draw the full structure. OH, and then we have again OH, we have again OH, we have H, and then we have H, we have OH, and then we have 1, 2, 3, 4. We have OH here, we have H here, and then we have H here. Okay, so this is galactose. In this case, it is again D because OH group is present on right hand side of this carbon next to CH2OH. Although this OH is present on left hand side, but this doesn't matter, we have to count only this carbon. Okay, in case of fructose, we have again OH, OH, and then we have H, OH, H, and H. Again it is D-fructose because again OH group is on right hand side of the carbon next to CH2OH. So this is important assignment to the carbohydrates D. Important to notice that all naturally occurring sugar are D in nature and none is found as a L. So this is a very important point to be remembered. And if we want to see asymmetric carbon atom, so we know that in glucose this is asymmetric carbon atom, asymmetric, asymmetric, asymmetric. So we have 1, 2, 3, 4 asymmetric carbon atom. So which one is asymmetric carbon atom? Carbon along which all the 4 groups are different. So those would be the asymmetric carbon atom. And we know that how many isomers are possible for asymmetric carbon atom? 2 raised to power n, where n is number of carbon atom. We have n is equal to 4 in case of glucose. So total 16 isomers are possible just by changing the position of OH and H left or right. Then we can make combination of 1, 2, 3, 4, then 1, 2, 1, 3, 2, 3, 2, 4, like that. So we will total get 16 isomer of glucose. Next is the preparation of carbohydrates. Let's say we want to make glucose and galactose. So we have molecule which is called lactose. So if we do hydrolysis of lactose, we get glucose and galactose. And in case of starch, if we do again acid hydrolysis in the presence of temperature and pressure, we get the glucose. So preparation is not that important, but we have the structure which is D configuration of glucose. This is very important. And further, D doesn't correspond to deoxyrotatory or levorotatory, which means that it can rotate the plane polarized light into left or right direction. This is not that D, that is represented by plus and minus. So this D is just left or right with respect to the OH group. Okay, so this is the important aspect of the structure of the carbohydrates. Now we have learned that there are 6 carbons in glucose. There is aldehyde group, there is hydroxyl group. But question is, how do we know that these groups exist? There is 6 carbon. How scientists came to know about all these things about the glucose molecule or carbohydrate molecule? So they did number of experiments, number of years were spent to figure out the structure of glucose, and ultimately they came up with the formula and structure. Now we will see those reactions which people did to confirm the structure of glucose. Okay, so next is various reactions which can predict or which showed us the structure of the glucose, how it comes up with the time. Okay, तो अभी हम देखेंगे various reactions of glucose which predicted how glucose molecule should be. For example, first is reduction in the presence of hydrogen and sodium amalgam. Because this is reduction reaction, it converted aldehyde group into CH2OH. Okay, because we know that sodium amalgam can reduce aldehyde group, from here we came to know that in this molecule, which was unknown at this time, contain one aldehyde group. Only then it can be converted into alcohol. So this was the first reaction to show that there is aldehyde group present. So next is HI. HI is a strong reducing agent. It reduced everything like aldehyde, OH, 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 OH. It reduced everything into carbon chain hexane. Okay, it did complete reduction. From here we came to know that this molecule, whose structure was unknown, contained at least 6 carbon. Only that we can obtain hexane molecule. Okay, after that is acetylation reaction. We know that acetic anhydride reacts with alcohol to form the ester. So there is alcohol group present. How many? 1, 2, 3, 4, 5. And this acetic anhydride, it formed ester like CO, ester. Okay. So from this reaction we came to know that this molecule has 5 hydroxyl group because of this ester formation in the presence of acetic anhydride. So up to this stage we came to know that this molecule, whose structure was unknown, contain aldehyde group, contain 6 carbon, contain 5 hydroxyl group. And then next reaction which was done by HCN. So in HCN molecule we know that it reacts with aldehyde. So here I have drawn aldehyde, C double bond O, O is electronegative, it has some partial negative charge, carbon is partial positive charge. In HCN, cyanide has partial negative charge and hydrogen is partial positive charge. So this negative ion, negatively charged moiety reacts with positive charge here, okay, and then this positive charge goes to the oxygen. So we get a molecule like C, and then we have OH, and then we have CN. Okay, so both molecule entity get added to the carbonyl group CN, H and OH. Because this HCN reacts with aldehyde, this further confirms that molecule has aldehyde group in it. The next reaction with NH2OH. Again we have C double bond OH plus NH2OH, and this NH2OH we can write in the form of H2NOH. From here we have removal of water molecule, okay, and then we get C double bond NOH. Because this is also a characteristic reaction of aldehyde group, we came to know that this molecule must contain aldehyde group. And then we have reaction with bromine. This acts as the oxidizing agent and convert aldehyde group into acid group, forming C double bond OH. And next we have reaction with oxygen in the presence of HNO3. HNO3 is a strong oxidizing agent. It oxidizes both aldehyde, terminal aldehyde and terminal alcohol group. So we get two carboxylic group, this, and then we have COH 4 times, and then again we have COH. Okay, so we get two carboxylic group with the nitric acid. And then we have reactions with Tollin's reagent, silver ammonium in the alkaline medium, and then we have copper plus 2 in the alkaline medium. The first reaction is called, this reagent is called Tollin's reagent, and it forms silver precipitate. So that's why it is called silver mirror test for the glucose. In case of copper plus 2 5 hydroxyl ion, we have copper 2 oxide, copper oxide. So this gives red precipitate, which is also a test for the glucose molecule. So with these all structures or all these reactions, it was concluded that structure of glucose should be something like that which we have drawn here. And after that there are more important reactions of the glucose, which is with phenyl hydrazine. It forms oxazone, which is having a yellow precipitates, and this is also very important reaction. So let's see what is the oxazone formation in the presence of phenyl hydrazine for the glucose molecule. Okay, so we have glucose molecule already here, and then we, let's say, plus phenyl hydrazine. So phenyl hydrazine is a molecule like NH2 NH C6H5, phenyl hydrazine. So in overall reaction, 3 molecules of phenyl hydrazine reacts, and out of 3, 2 molecules get added to the glucose molecule. Okay, so ultimately product we will have C 1 2 3 4 5 6, okay, C double bond, because in this case we have H2N NH phenyl group. This water molecules get removed again, so we have C double bond N NH and phenyl group C6H5. So one molecule reacted with aldehyde group. Now second molecule comes. The function of second molecule is to oxidize this alcohol into ketone group. Okay, so second function of second molecule of hydrazine is to convert alcohol into ketone group, and then that ketone group reacts with third molecule of phenyl hydrazine to form osazone. Phenyl hydrazine, and react structure is same as glucose. Okay, so 2 molecules get added but 3 are consumed, and this molecule is osazone, and it has yellow precipitates, yellow color, and that's why it is also important reaction and can be used for the as a test. Important question is from this is that although only 2 molecules are added but 3 molecules are used, explain why. As I already mentioned, second molecule is used to convert alcohol into ketone group C double bond O, and then that C double bond O ketone group reacts with third molecule of phenyl hydrazine. So that's why 2 are added but 3 are consumed. So this is also very important reaction of glucose molecule. Okay, so after that we will see further about glucose molecule, and let's see what is the next topic in this category. तो अभी हम करेंगे साइक्लिक स्ट्रक्चर ऑफ ग्लूकोज। अभी हमने ये तो देख लिया कि ग्लूकोज की स्ट्रक्चर में क्या क्या फंक्शनैलिटी प्रेजेंट है, जैसे कि एल्डिहाइड ग्रुप है, अल्कोहल ग्रुप है, 6 कार्बन है, ये सब हमने उसके कैरेक्टरिस्टिक फीचर से या कैरेक्टरिस्टिक रिएक्शन से देख लिया। लेकिन इन सभी के बावजूद भी देयर वर सम रिएक्शंस, देयर वर सर्टेन थिंग्स व्हिच कुड नॉट बी एक्सप्लेन्ड ऑन द बेसिस ऑफ लीनियर स्ट्रक्चर ऑफ द ग्लूकोज। फॉर एग्जांपल this structure which we call as a linear structure. So some of the characteristic reactions of aldehyde or other properties could not be explained on the basis of linear structure. So we were in need of something else which could explain all the features. For example, it was found that glucose exists in 2 forms, alpha and beta, with different value of optical rotation. One is 110 and another is 19.2. And further it was found that when one form of glucose is kept in water, it gets converted into other form. So its optical rotation value changes with time until an equilibrium is achieved. For example, if I put alpha form in water, it will change 110 to 50.2, or if I put beta 19.2 degree optical rotation, it will change to 50.2.5. So it will, there was change in the optical rotation with time until it achieved an equilibrium, and this phenomenon is called mutarotation, where muta corresponds to mutation and rotation corresponds to optical rotation. But these concepts or these phenomena could not be explained on the basis of linear structure of the glucose. And then other structure was proposed, which was the cyclic structure. So what happens in the cyclic structure? Again, this is very important. So we have 6 carbon, 1, 2, 3, 4, 5, 6 of the glucose. This is aldehyde group at first position, and this alcohol group at carbon number 5. So see, this oxygen negatively charged partial, carbon is positively charged partially. In this OH group, this oxygen is partially negatively charged, this hydrogen is partially positively charged. What happens is that this OH group reacts with this carbonyl group here. Okay, this H goes to O and this oxygen goes to carbon. So we have a linkage carbon, oxygen, carbon, and H, which are present here in the form of OH, gets attached to the oxygen of the carbonyl, forming the OH, and this forms a cyclic structure. Important thing to note is that what position of carbon is involved? 1 and 5. This is important. That linkage is formed between carbon 1 and carbon 5. So this was a cyclic structure, and which can be further represented as in the form of this pyranose ring. So in general, how to draw this structure? So you can first draw simply a pyranose form, okay, like this, and just put one oxygen on the top right, okay, and all the position draw vertical lines, okay. On top left always write CH2OH and then H, okay. Now OH is on upper side, here OH is on downside, OH is on downside, OH is on upside, OH is on upside, OH is on downside in case of glucose. And now OH is on downside, OH should go upside, but we are going to draw alpha form. In alpha form, OH is always on downside. Okay, so you can remember in this way. I will repeat. First, draw the structure of pyranose like a hexagon. On top right, always put oxygen. Then put vertical lines at all the positions. On top left, always write CH2OH and then H. Now OH is on upside, here is OH is on downside, downside, upside, upside, downside, and then it should be upside, but because we are going to write alpha form, and remember in alpha form OH is always on downside, so here it is again downside. Now if we have to write beta form, just we have to convert alpha to beta, OH downside to OH upside. Other things remain the same. Again draw a hexagon, here oxygen, always draw vertical lines, CH2OH always on top left, OH is upside, downside, upside, downside, because it is beta form. In beta form OH is always on upside. So this is a cyclic form of the glucose, and this is very important. Similarly, we can draw the structure of fructose molecule also. We have seen fructose of glucose, now fructose, structure of fructose. Again we have 1, 2, 3, 4, 5, 6 carbon, and linkage is supposed to form between carbon 2 and 6. This is again important. Here we have drawn the linkage between 1 and 5, now it is 2 and 6. Again 2 and 6. This OH goes to this carbon forming a oxygen linkage. Okay? And from this again we can draw the pyranose form. Again draw a hexagon with O on top right. Okay, and then draw all the vertical lines. Okay, now in this case this is CH2, so your carbon 6 has HH. It's a little bit different compared to this one, because at last position we had CH2OH, but here in last position we have CH2, so HH. Okay, next read or compare with respect to glucose. OH downside, OH downside, H up, OH up, OH down, OH down. Alpha form, OH is always downside, OH down, OH down. Compared to H we have CH2OH. So in other words, all these positions remains the same. Okay, only this is changed here. Okay, in other words you can remember this CH2OH group from here is gone to here. This CH2 group जो कि ग्लूकोज में यहाँ पे था मूव होके वहाँ पे चला गया. So this form structure become your fructose form, which is the alpha form. Okay. It is important that fructose in free state exists in 6-member ring, but fructose in combined form, like in case of disaccharides, polysaccharides, it exists in 5-member ring rather than the 6-member ring. Okay. In case of 5-member ring, we have furanose form, oxygen, again draw the vertical lines. Okay, CH2OH, CH2OH, both are same here. Okay, and then because we are drawing alpha form, OH is always downside. Okay, downside, upside, upside, downside, and then there is only H. So in this way you can just remember a little bit that what was the position of H and OH. And the most important is that you can remember the structure of glucose only, and from this you can derive the other forms. Alpha always OH downside, beta always OH upside. In case of fructose, draw same structure, okay, but just change the position of CH2OH from here to here and H go from here to here. This will become your fructose form in the pyranose form. Now if you have to draw fructose in furanose form, you have a pentagon with oxygen, CH2OH, CH2OH, same. It is alpha form, OH is always down, down, up, down. Okay, so this will be your alpha form of the furanose. Got it? So this was the cyclic structure in case of the monosaccharides. After that we will see some structures of oligosaccharides like disaccharides and then polysaccharides. In those forms always cyclic structure will be used and position of the linkage would be shown. For example, whether carbon 1 is involved in bond formation or carbon 4 is involved in bond formation. So in those cases, if you remember these structures, only then those structures can be remembered easily, otherwise it will become difficult. Okay, so after that we will do the structure of some disaccharides, and that topic is also very important for your competition point of view. Okay, तो अभी हमने मोनोसैकराइड्स के बारे में देख लिया, जान लिया कि वो कैसे होते हैं, उनकी स्ट्रक्चर क्या है. तो अभी हम देखेंगे disaccharides and polysaccharides. So in case of disaccharides, first example we have is the sucrose. Sucrose is made up of two molecules, glucose and fructose. So I have drawn the cyclic form of glucose and cyclic form of the fructose. We will see one of the difference between these two, how to draw this structure when we are going to make a disaccharide. Glucose is as simple as we learned earlier. Hexagon ring, oxygen, CH2OH, OH on, top, down and then down because it is alpha. In case of beta fructose, we have learned about this structure, CH2OH, then here is H, OH, OH, CH2OH and OH. This is carbon number 1, 2, 3, 4, 5 and 6. But here I have inverted this structure from here to like this. It was on plane like this, I made like this. So that carbon 2 position, this position is oriented towards the glucose. This position here. Okay, because this is beta form, in beta OH is always up, so this is upside, CH2OH downside. Now this OH and this OH, they are react and remove the water molecule, and glycosidic linkage is formed between the two. So one glycosidic linkage is formed between the two. So this structure is become structure of sucrose, and it is called alpha D glucose is involved and beta D fructose is involved, and this linkage is named as alpha 1 beta 2 linkage. Means alpha of carbon 1 and beta of carbon 2 is involved in the linkage. It is alpha 1 beta 2 linkage, and this is again very important because questions are asked what type of linkage exists in this molecule. Now maltose, it has glucose, both units are same, linkage is alpha 1 to 4, means one carbon of one glucose and fourth carbon of another glucose, and both glucose will be in the alpha form. So alpha 1 to 4 linkage. Lactose, we have glucose and galactose. In this case both glucose and galactose are in beta form and linkage form is beta 1 to 4. One carbon of glucose and fourth carbon of the galactose is involved in the linkage. Then we have polysaccharides. It has starch, amylose and myelopectin. In case of amylose, we have alpha 1 to 4 linkage. Everything is made up of glucose. In case of myelopectin, we have alpha 1 to 4 linkage from the glucose, and then alpha 1 to 6 linkage as well. How this 1 to 6 linkage is formed? When alpha 1 to 4 linkage is formed, a chain is formed, like one glucose molecule, another glucose, another glucose, another glucose, another glucose. So in this case we have alpha 1 to 4 linkage, alpha 1 to 4, alpha 1 to 4, alpha 1 to 4. This is one linear chain. Now there is another linear chain of same alpha 1 to 4, alpha 1 to 4 linkage. This is another linear chain. Now these two linear chains are parallel to each other. Okay. Now these two linear chains are held together by alpha 1 to 6 linkage. Okay. These two parallel chains are held together by alpha 1 to 6 linkage. Like there is a covalent bond between the two. and this happens in case of amylopectin. Next polysaccharide is cellulose. It has glucose but in the beta form, so linkage is beta 1 to 4. Both glucose are in beta form and carbon 1 and carbon 4s are involved in the linkage. This is a very important topic. Two type of questions can be asked. For example, in case of lactose, what are the constituent particles or constituent sugars, glucose and galactose? What type of linkage exists? Beta 1 to 4. Which carbons are involved? Carbon 1, carbon 4. Such type of questions can be asked. And also for the what is the linkage between two parallel chain of carbohydrates in case of amylopectin, that is alpha 1 to 6 linkage. Another important questions can be asked about invert sugar and non-reducing sugars. So what are the invert sugar? Sucrose is known as invert sugar because sucrose has optical rotation in plus, okay, but when it get hydrolyzed into glucose and fructose form, its optical rotation changes from plus to minus because fructose has more contribution towards the minus optical rotation. Okay, so that's why it is called invert sugar. Sucrose plus, after hydrolysis mixture is minus. Sucrose is also called non-reducing sugar because it cannot reduce Fehling solutions or tolerance reagent. It doesn't react. Why? Because in case of sugar we have linkage carbon 1 and carbon 2. These both carbons, which were carbonyl carbon, are involved in glycosidic linkage. In other words, no free aldehyde carbon or ketone carbon is present in case of sucrose. That's why it is called non-reducing sugar. But in case of glucose like maltose, lactose, etc., one free carbonyl carbon is present, so that's why they are called a reducing sugar. You can see linkage is 1 to 4. One is the carbonyl one, this get used up, but another sugar used fourth carbon. It doesn't use its first carbon, which was a aldehyde group carbon. So this is free in this case. It is reacting. So that's why it is reducing sugar. Similarly, in case of lactose, 1 to 4 first carbon, fourth carbon, but that first carbon of another sugar is free. So that's why they are called the reducing sugar. But in case sucrose, both 1 and 2 carbons are involved, so it is non-reducing sugar. So there are some important questions: what type of linkage, what type of molecules exist between various disaccharides or polysaccharides, and question can be asked. With this, I think we have finished our carbohydrates part, and next we will see proteins and DNA. Okay. तो अभी हम करेंगे अमाइनो एसिड एंड प्रोटीन्स. अमाइनो एसिड्स, there are 20 amino acids which exist in nature and they are classified as a basic, acidic and neutral depending upon what type of R group is there. And they are also further classified into essential and non-essential amino acids. Essential amino acids which are usually asked in competition exams are VIAL, LH, MP, TT. So how you can remember VIAL as a vial? Okay, you know what is vial? Like these small glass vials which are present usually in the lab to store the samples. Vial. LH, you can remember from Ludhiana, LDH as abbreviation, L and H from Ludhiana. MP as a Madhya Pradesh, TT as a Telangana, Tamil Nadu, Tripura, whatever you want to say. So this essential amino acid can be remembered from this formula: vial, Ludhiana, Madhya Pradesh, Tripura, Telangana or Tamil Nadu. Okay, so in completion this question can be asked out of the following which one is the essential amino acid. And general structure of amino acid is that we have C, one acidic group and one amino group, and then R and H. Now again, as we did in case of carbohydrates, if OH group is present on right hand side, it is designated as D. If it is present on left hand side, it is called as a L. Similarly, in case of amino acid, If NH2 group is present on left hand side, it is called N, and if it is present on right hand side, it is called D. Contrary to the carbohydrates, in amino acid configuration is always L, that is NH2 group is present always on the left hand side. So all natural occurring amino acids are L in form and all carbohydrates are D in form. So in terms of structure, amino acid exists as zwitter ion. In this case, H from the acetate or carboxylic group gets shifted towards the NH2 group, making NH3 positive and CO negative. So this is a structure which is found in case of amino acid and is called zwitter ion structure. Okay. Isoelectric pH. So as this ions has some charge, it has some tendency to move under influence of electric field. Okay. So depending upon pH, either they will move towards cathode or anode. But at the pH, when charge is neutral, there is no net movement, there is no net migration of the amino acid towards either electrode. We call it as isoelectric pH. So pH at which there is no net movement, we call as the isoelectric pH. What are the peptides? So when two amino acids combine, it forms a peptide linkage, and that is called peptide. So if two amino acids are, then dipeptide, if three, then tripeptide, if four, then tetrapeptide, and then as many as polypeptide. So what happens is NH2 is on left hand side, COH right hand side, this OH and this hydrogen from the NH2 group removed as a water, and we get here as a C double bond O NH group. This linkage is called the peptide linkage. Okay, so if there are another amino acid, we will get tripeptide also. By convention, NH2 is always kept on left hand side. When a protein is formed, in protein there would be many amino acids like 1,000 and more. So we call it as N terminals of protein and C terminal of the protein. It is understood that if we are saying N terminus, so we are talking about this end, and if we are talking about C terminus, we are talking about this end of the protein. And this is very common nomenclature used in the study of proteins: N terminus end or C terminus end. So this was the basic about amino acids and their structures, the classification, and the peptide bond formation. When we have peptide, then after that we go for the protein. Okay, because we know that proteins building blocks are amino acids, and amino acid combines to form di, tri, tetra, penta, hexa and polypeptide, and when polypeptide is formed, a protein is formed. Although there is no any sharp boundary, any sharp demarcation between difference between polypeptide and proteins, but it is said that if molecular weight is less than 10,000, then we say polypeptide, and if molecular weight is more than 10,000, we say it as a protein. Now we will go for the structure of the proteins as a primary structure, secondary structure, tertiary and quaternary structure of the proteins, and then move forward towards the enzyme also. Okay, तो अभी हम देखेंगे structure of proteins. So first we have primary structure. In primary structure, there is just a sequence of amino acid in particular order. Like it may be valine, alanine, leucine, isoleucine, tryptophan, methionine, like that. And this is one of the most important, very crucial part of protein structure. In case even a single amino acid is changed in the sequence, it can drastically change the function of the protein, or even it may make it completely inactive. For example, one example is hemoglobin. If one amino acid GLU, glutamine, is changed to valine, which is a basically mutation, which means change, function of protein or shape of protein is definitely changed. In this case, hemoglobin RB cell become sickle cell compared to their spherical shape. Spherical to sickle cell. Okay, so this one change, rather only change of one amino acid has changed the shape of the cell, and it has some side effect on humans. So primary structure, which is sequence of amino acid, is very important. Then we have secondary structure, which is alpha helix and beta plated. In alpha helix we have some helix formation like this, okay, and then there are some amino acids, okay, and in this amino acid there is hydrogen bond, okay, and it has been observed that in this amino acid after every 3.6 amino acid hydrogen bond exists, okay. And in secondary structure it is very important that rigid nature of the amide bond. We have learned that in amino acid when peptides are formed there is amide bond formation, and this amide bond has rigid nature. And how it comes up? We have C double bond O and H. This lone pair of electron goes towards this carbon, form a double bond, and this shifted towards the oxygen. Now this is C double bond N, across which rotation is not possible, so that's why it become rigid in nature, and this is very important aspect for your competition exam, okay. And this comes in secondary structure of the protein. And alpha helix, and then we have the beta plated sheet, which has parallel and anti-parallel sheets, which we have already discussed. One thing, that when peptide is formed it runs from N terminus to C terminus. So when two sheets, one sheet and two sheets, they are running parallel in the same direction, N to C, N terminus to C terminus, N terminus to C terminus, it is called parallel beta plated. But when it is anti-parallel, like N terminus to C terminus, but next sheet is C terminus to N terminus, N to C, but next is C to N, so then we call it as a anti-parallel beta plated sheet, and both the sheets are held together by hydrogen bond like a weak forces. After that we have tertiary structure. In tertiary structure, when primary structure is formed, secondary structure is formed. In tertiary structure, those structures are further get folded in three-dimensional form to give a new conformation. Then we call it get a tertiary structure. And this is a structure which is important for functioning of the proteins or the enzyme. Quaternary structure, when such a tertiary structure polypeptides combine further, like 1, 2, 3, 4, when they combine further by weak van der Waals forces or hydrogen bonding, then we get a quaternary structure of the protein. So in some cases such type of structure are important for the functioning of the protein. So these are the some various steps by which a protein get particular conformation. Primary, sequence of amino acid; tertiary, in form of alpha helix and beta plated, and then secondary, and then tertiary, further folding in 3 dimension, and quaternary, when 2 or 3 molecules of tertiary structure further combines to give another molecule, then it form a quaternary structure of the protein. So in case any change happens at any stage, protein function can get damaged or drastically changed, okay. After that we have another next topic is the enzyme. We will see that one. Next we will see about enzyme. Enzymes also known as biological catalysts. So they are very vital in our body because a number of reactions which are catalyzed by enzymes are very fast. If we eat our food, it may take many years to digest single meal, but in the presence of enzyme it get digested in just couple of hours. So they are the biological catalyst and they accelerate the rate of reaction quite fast. What are characteristic feature of enzyme? They are highly efficient. Efficiency is very high because they accelerate the rate of reaction manifold, like 100,000 folds. They are needed in very small quantity, so very little amount is sufficient to do the reaction. They are very specific in nature, means a particular enzyme can catalyze a particular substrate only, it will not react on other substrate, and they have particular optimum temperature and pH at which they can work. If temperature and pH is changed, they will not work to their highest efficiency or highest specificity. Some of these terms, like they are specific in nature and they are highly efficient, these are something like old aspect of the enzyme. But today, as we have more knowledge of the enzyme, as we are doing more research, the specificity is not that much true for the enzyme. It has been found that one enzyme can act on different substrate also. So in this case, enzyme lose its specificity. In terms of efficiency, certain enzymes are extremely slow. They can do some reaction definitely, but rate of reaction is not that fast. So they are not that efficient also. But in major cases, these parameters holds true. But in present time, as we have gained more knowledge about the enzyme, some of these are not that much true. What are the cofactors and coenzymes? So sometimes enzymes cannot work by its own. What are enzymes? They are made up of proteins like amino acid sequence. But for some cases, we need additional factors which might be covalently bound to the enzyme. In that case, it is called cofactor when they are covalently bound. And when they are not covalently bound or just come into the picture when reaction is going to happen, they are called coenzyme. When reaction is over, they may get removed. What is the mechanism of enzyme reaction by which they work? It is based on lock and key hypothesis. Means, for example, there is some particular shape of the enzyme. In this, only a particular substrate can fit. Lock and key. Like a particular key can fit in a particular lock. Similarly, particular substrate can fit into particular enzyme. So when this lock and key has fit enzyme, substrate has fit into the enzyme, its activation energy get lowered, so it makes enzyme substrate complex. Okay, then it form a product here, enzyme product, and then enzyme get separate and product get separated. Product and enzyme. So this is a mechanism of enzyme reactions. Enzyme substrate complex, activation energy is lowered, enzyme product is formed, enzyme is separate and product is separate. Next is hormones. What are the hormones? Hormones are the chemical messengers, and mostly they are secreted by pituitary gland, which is present near the brain, and pituitary gland is also called the master gland of the body. So they are chemical messengers. So they are secreted from pituitary gland and they act on another gland and starts a cascade of reactions, and then a number of metabolic processes starts. So they basically give a signal to start a metabolic process. So that's why they call the chemical messengers. And what are the classification? They are steroidal in nature. For example, all sex hormones are steroidal in nature. Proteins like oxytocin, vasopressin, insulin, they are proteinaceous in nature. And then amines like adrenaline and thyroxine, they are amine in nature. So each hormone has its own specific function in the body. If something is disturbed in our body, something is perturbed, this metabolic processes can also get disturbed if hormones get disturbed somehow. Okay, so this was something about enzymes, their mechanism and the hormones. And next we will proceed towards the vitamins and further more topics. Okay, so let's go ahead with the description of vitamins. Vitamins are initially known as vital amines, but later the idea was dropped because all vitamins were not the amine. So they are classified as fat soluble vitamin, which are A, D, E, K, and water soluble vitamin, which are B and C. So A is another name is retinol, and if it's deficiency, it causes night blindness and xerophthalmia. Next is vitamin D, another name is calciferol. It causes rickets and is important for development of bone and teeth. Next, E, which is also known as tocopherol. It's important for sexual power and muscle fiber formation. Vitamin K, also known as phyloquinine. It is important factor for coagulation, that is blood clotting. If it is deficient, a person may bleed more than the normal person. Vitamin B1, which is thiamine, it causes a disease called beriberi. Vitamin B2, it is called riboflavin, and it can cause skin diseases if there is deficiency. Vitamin B6, which is pyridoxine, it is important for nervous disturbance. Vitamin B12, which is cyanocobalamin, it can cause anemia if there is deficiency. And then vitamin C, which is ascorbic acid and mostly obtained from citrus plants, and it can cause scurvy if it is deficiency is there in the body. So important question which can be asked from this is another name of vitamin. Let's say vitamin E is also known as, what is the option? Tocopherol. Vitamin B is also known as thiamine. Or what type of disease it can cause, or why it is important for the body, like what are their functions. So these two type of questions can be asked from the vitamins. Okay. After that, our last topic is nucleotides, DNA, RNA and the basic units. We will do that. तो अभी हम करेंगे अपना last topic of the chapter, that is nucleic acid. So we are well aware of the two things, that is nucleic acids are DNA and RNA, and they are called polynucleotides because they are made of nucleotide, which is their basic unit. And poly means many, so there are many nucleotides in a DNA or RNA. Now each nucleotide is made up of three parts, that is nitrogen base, sugar and phosphate group. In nitrogen base we have two type of nitrogen bases, purine and pyrimidine. In purine we have two further AG, adenine, guanosine, and in case of pyrimidine we have three, thymine, cysteine and uracil. Important thing to note is that in case of DNA we have A, G, T and C, but U is absent. But in case of RNA we have AG and UC, but T is absent. So this makes one of the major difference between DNA and RNA. Then we have sugar. Sugar is present in both DNA and RNA. In DNA it is deoxyribose sugar, and RNA it is ribose sugar. So again this is one of the difference between DNA and RNA. Deoxy means removal of oxygen, so one oxygen is missing. In case of RNA it is complete ribose sugar. And another important thing is that both the sugar are in beta form. So we have learned earlier that there are alpha and beta, two forms of the various sugars in case of carbohydrates, like alpha D glucose, beta D glucose, and in case of DNA and RNA we have beta ribose sugar. Okay. Now these three things, nitrogen base, sugar and phosphates, are combined to form the nucleotide. And how these are combined? So this in the center I have drawn a sugar, and it is a, let's make it a D-ribose sugar. At 2 position there is only H. If it is ribose sugar, at 2 position there would be OH, because I am talking about DNA, so it is deoxyribose. So at 2 position oxygen is gone. Now this is first carbon. As I mentioned, it is beta ribose sugar. So in case of beta we know that OH group is always up. So this was the OH. Now this OH has made a bond with the nitrogen base, which may be Ag, T or C. So first carbon OH is involved in the bond formation with the nitrogen base. Now we go from 1, 2, deoxy, 3, 4 and 5th carbon. At 5th carbon we have phosphate group attached. Okay. So this whole, when we have phosphate, ribose and nitrogen, all these three combine, and then we call it as a nucleotide. Now let's say there is another unit of nucleotide. So again we have sugar. Let's make this deoxyribose sugar. Oxygen is missing at carbon 2. This is carbon 1 OH. It is on upside because sugar is beta, and this O is involved in bond formation with nitrogen base. Now 1, 2, 3, 4 and 5th carbon, there was phosphate group present, but in this case this phosphate is involved in bond formation with hydroxyl group of 3rd carbon of another nucleotide, and this bond formation is called phosphodiester bond, which makes the backbone of the DNA. Important thing in this structure is to remember which position is attached to which position. I will repeat again. 5th carbon OH, this is involved in phosphodiester bond formation with OH of third carbon of another nucleotide. And at the end of the DNA, this 3-hydroxyl group remains the free because it is not further involved in any bond formation. Okay. And another aspect of DNA structure is that it is negatively charged because of the presence of phosphate group. So DNA is negatively charged species. Okay. So this was all about basic unit, which is a nucleotide, phosphate, sugar and nitrogen base. Now if I remove phosphate group from this whole nucleotide, then we call it as a nucleoside. That is only sugar and nitrogen base, then it is called nucleoside. Okay, so this was the basic unit, and then let's see the structure of DNA and RNA. So structure of DNA, it is double helix structure like this. There are two helix run anti-parallel to each other, and in double helix structure in DNA, adenine is also A is always combined with T, adenosine always combined with thymine, and cysteine always combined with guanosine. So this is the by rule in the nature that A always combines with T and C always combines with G. 2 lines means there are 2 hydrogen bond between A and T, and 3 lines means there are 3 hydrogen bond between C and G. So bonding between C and G is more stronger compared to the bonding between A and T. Okay, in case of RNA, instead of double helix we have single helix, and A always combined with U because we know that in RNA T is missing, and C always combines with G, same as in case of DNA. Okay, now in case of DNA there are 2 strands which are complementary to each other, and they run anti-parallel. So what does mean complementary? For example, if I have the sequence of one strand, let's say I know that there is A, there is T, there is C, there is G, I automatically know the sequence of other strand. This would be T, this would be A, this would be G, this would be C. Okay, so if I know the sequence of one strand, sequence of another strand is automatically get fixed, because we know that A can always combine with T and C can always combine with G. So that's why we say that 2 strands are complementary to each other. And those strands are anti-parallel also, means one strand runs from 5' to 3' direction, and other strand run from 3' to 5' direction, and these directions comes from here, which I already mentioned, 3 hydroxyl group and 5 hydroxyl group. So that's why we call it as anti-parallel strand. So these are some of the characteristic features of DNA, okay, which are very important, and sometimes questions are also asked. Now next important aspect of DNA is its replication. Replication means DNA has tendency to produce its own duplicate copy, and then we call it as a replication of DNA. So this was the DNA in double helix form, as such intact. Now when enzymatic process starts, DNA replication starts, these 2 strands gets opened up, okay, hydrogen bond between the 2 gets broken up, 2 strands get opened up, and new strand starts forming. Okay, new strand starts forming with the help of enzyme DNA polymerase. So this is DNA replication, and it is called semi-conservative in nature. Why it is called semi? Semi means half. So half means one strand of newly formed DNA. This is a newly formed DNA. One strand of newly formed DNA belongs to the previous strand, okay, and this strand is the new strand. So I can say half of the DNA strand was conserved, because half is coming from the previous generation and half is coming from the new generation. So I can say it is semi-conservative in nature, that is half is from previous and half is from next generation. And DNA replication proceeds from 5' to 3'. And this is again very important aspects for the competition exam. That is, DNA replication proceeds in which direction? 5' to 3'. What type of mode is there in DNA replication? We say it is a semi-conservative nature. In structure of DNA, two strands are complementary and run anti-parallel to each other. Okay, so with this we have finished our chapter biomolecule. So we covered various biomolecules like carbohydrates. Among that we had monosaccharides, oligosaccharides, polysaccharides, and then we covered some amino acid proteins. Then we covered hormones, vitamins, and ultimately to the DNA, RNA. Okay, so I believe that with this much knowledge you would be able to you have sufficient knowledge to attempt some of your important questions from your competition exam. And now we will proceed to some examples from your question booklet. Okay, तो अभी हम करेंगे कुछ क्वेश्चन बेस्ड ऑन बायोमोलिक्यूल्स. So your first question is general formula of the carbohydrate is, and your options are CnH2n O2n plus 2 CxH2O 2x CxH2OY and then none. So the question is general formula of the carbohydrate is. So we know what is general formula of carbohydrates, that is hydrates of carbon, and we know that it's X and Y value. So answer is C. Okay, so next is optical activity is shown by. So your options are glucose, fructose, sucrose and all. So question is optical activity is shown by glucose, fructose, sucrose, all. So what would be the answer? It is all, because in each case we have the chiral carbon present. So if there is chiral carbon present, so the compound will show the optical activity. Okay, next question is ultimate product of oxidation of most of hydrogen and carbon. So क्वेश्चन क्या है कि अगर हम किसी भी कार्बन अल्टीमेट प्रोडक्ट ऑफ ऑक्सीडेशन ऑफ मोस्ट ऑफ हाइड्रोजन एंड कार्बन इन फूड स्टफ, मीन्स फूड स्टफ में जो भी कार्बोहाइड्रेट्स वगैरह हैं उनको अगर हम अल्टीमेट ऑक्सीडेशन कर दें तो उसके प्रोडक्ट्स क्या होंगे? वाटर एलोन, कार्बन डाइऑक्साइड एलोन, नेक्स्ट इज वाटर एंड कार्बन डाइऑक्साइड एंड नेक्स्ट इज नन. Okay, so if we do continuous oxidation for any carbohydrates or any biomolecules, carbon will be always converted into carbon dioxide and hydrogen will always convert into water. So answer would be water and carbon dioxide. Okay, so let's say next question. Which of the following foodstuff contains nitrogen? Which of the following foodstuff contain nitrogen? Options are carbohydrates, fats, protein. Options are carbohydrates, fats and protein. So which out of these contain nitrogen? We know that protein, because it has amino acids. So in amino acid we have the nitrogen. So answer is protein. Okay, so let's say next question. Nucleic acid is a polymer of nucleosides, nucleotide. So question is nucleic acid is a polymer of nucleosides, nucleotides, alpha amino acids and bile acids. So nucleotide is polymer of which one? Nucleotide. Okay, so because we know that in nucleotide we have ribose sugar, nitrogen base and phosphate group. So all three are required and that is the polynucleotide. Answer is nucleotide. Okay, next is secondary structure of protein refers to? Next question is secondary structure of protein. It refers to what? Alpha helical, hydrophobic interaction, sequence of alpha amino acid, sequence of alpha amino acids, fixed configuration of polypeptide backbone, fixed configuration of polypeptide backbone. So what is secondary structure of protein? So we know that primary structure is just sequence. So this is primary. Polypeptide backbone is just the formation of C double bond ONH, and hydrophobic interaction is just a force of attraction. So answer left is only alpha helical as a secondary structure, in which other form is beta plated. Okay, so let's see next question. Which of the following is not a fat soluble vitamin? Which of the following is not a fat soluble vitamin? Vitamin B, D, E, A. Vitamin B, D, E and A. Not fat soluble. If it is not fat soluble, it means it is water soluble. If it is water soluble, then answer is B, and other option is vitamin C, that is also a water soluble vitamin. Okay, let's see. Deficiency of vitamin B1 causes the disease. So vitamin B1 की deficiency से कौन सी disease होता है? So your options are, let's say A, convulsion, and then you have beriberi, and then sterility, and one is callosis. Okay, so the question is, deficiency of vitamin B1 causes which disease? The answer is beriberi. Okay, let's see another question. Which hormone is produced under the condition of stress which stimulate glycogenolysis in the liver of human? So basically, which of the following is a stress hormone? So hormone which is produced under stress condition is your stress hormone. Option are thyroxine, insulin, adrenaline, estradiol. So your options are thyroxine, insulin, adrenaline, estradiol. So which of the following is a stress hormone? Answer is adrenaline. This is also known as stress hormone. Okay, so now we have seen some couple of questions also from your chapter biomolecule. The rest you can do at your home and attempt to do. And good luck with your exam. And with this we finished our chapter biomolecule with its theory and questions. Thank you.